As you settle into this moment, feel the gentle weight of your body wherever you rest. Notice your breath, moving in and out without conscious thought, without effort. Each breath brings oxygen to your cells, and with that oxygen comes something else. Countless microscopic particles from the world outside, particles that your body encounters, recognizes, and responds to with extraordinary precision. This recognition, this response, this quiet protection happening right now inside you is the work of your immune system. Tonight we explore immunology, not as a catalog of cells and molecules to memorize, but as a living story of protection, recognition, and balance that has been unfolding inside you since before your first breath. We will move slowly through this inner world, discovering how your body distinguishes self from other, how it remembers past encounters, how it maintains peace with beneficial organisms while defending against genuine threats. We will see how the immune system is not a simple barrier, but a sophisticated network of communication, learning, and adaptation. Immunology is the study of how living organisms protect themselves from infection, disease, and foreign substances. It asks fundamental questions. How does your body know what belongs and what does not? How does it remember past infections and respond more quickly the second time? How does it coordinate responses across different tissues and systems? And perhaps most remarkably, how does it maintain tolerance to your own cells, to food, to beneficial bacteria, while remaining vigilant against genuine danger? The answers to these questions reveal something profound about life itself. Every human being carries within them an immune system that is at once ancient and new. Ancient because its basic mechanisms have been refined over millions of years of evolution. New because it actively learns and adapts throughout your lifetime based on your unique experiences with the microbial world. The immune cells developing in your bone marrow right now are being educated to recognize the specific version of self that is you, different from every other person who has ever lived. This is not a system you can see working, yet it works constantly. While you think and move and rest, while you eat and breathe and heal small injuries you never notice, your immune system conducts an ongoing conversation with your body and with the world. It is reading molecular signatures on every cell it encounters. It is releasing chemical messages that coordinate responses across vast distances in the body. It is creating memories of past encounters that may protect you decades into the future. So as we begin this exploration, let your mind rest in the knowledge that we are learning about something both intimate and magnificent. The immunology we will explore is your immunology. The processes we will understand are happening inside you now as your innate immune cells patrol your tissues, as your adaptive immune cells circulate through your blood and lymph, as your body maintains the delicate balance between protection and tolerance. Let us begin gently, not with definitions, but with an image. Imagine your body as a vast city. This city has borders and checkpoints, internal security and defense forces, communication networks, and memory archives. The immune system is not a wall around the city, but rather an intelligent, distributed network of recognition and response that operates at every level, from the surface barriers to the deepest tissues, from immediate reactions to carefully coordinated campaigns that unfold over days and weeks. The immune system has two major divisions, often called innate immunity and adaptive immunity. These names themselves tell a story. Innate immunity is what you are born with. the immediate, fast-acting responses that recognize general patterns of danger. Adaptive immunity is what you develop through experience. The slower, more specific responses that learn from each encounter and create lasting protection. These two divisions work together seamlessly, constantly communicating, each enhancing the other. Innate immunity responds within minutes to hours. It includes physical barriers like your skin and mucous membranes, chemical barriers like the acid in your stomach, and antimicrobial peptides on your surfaces, and cellular defenders that can recognize and respond to broad categories of threats. These innate mechanisms do not need prior exposure to work. They are ready from birth, encoded in your genes, waiting to respond to patterns that evolution has recognized as danger over countless generations. Adaptive immunity takes days to weeks to fully develop, but creates exquisitely specific responses and lasting memory. It involves lymphocytes, T cells, and B cells that can recognize individual molecular structures with extraordinary precision and generate responses tailored to specific threats. This is the immunity that improves with experience, that makes your second encounter with a pathogen less severe than your first, that allows vaccination to work. Between these two systems flows constant communication. Innate immune cells do not just fight, they also inform. They send molecular messages to adaptive immune cells, describing what they have encountered, activating specific response programs. Adaptive immune cells, once activated, send their own signals back, enhancing innate responses, coordinating the overall defense. The immune system is not a hierarchy with one part commanding another, but rather a network of conversation, each component influencing others, the whole emerging from countless local interactions. Before we can fully appreciate how this system works, we need to understand what it works with, the fundamental building blocks of immune function. At the most basic level, immune responses depend on recognition. The immune system must distinguish between what is self, your own cells and molecules, and what is non-self, foreign substances, microorganisms, altered or damaged cells. This recognition happens through molecular interactions, specific shapes fitting together like keys and locks. On the surface of every cell in your body are molecules that announce identity. These are the major histocompatibility complex molecules, often called MHC molecules. Think of them as molecular identification badges that display samples of what is happening inside the cell. If a cell is healthy and normal, it displays normal self-proteins. If a cell is infected by a virus or has become cancerous, it displays abnormal proteins. Immune cells constantly read these displays, checking that everything is as it ought to be. The immune system also recognizes molecules that are universally associated with danger, patterns found on bacteria but not on human cells, molecular signatures of cell damage or death, substances that indicate tissue injury. These are detected by pattern recognition receptors, proteins that have been shaped by evolution to identify the molecular structures common to threats. When these receptors bind their targets, they trigger signaling cascades inside immune cells, activating defensive responses. Recognition alone is not enough. The immune system must respond appropriately, not too weakly to allow infection, not too strongly to damage healthy tissue. This requires coordination across multiple cell types, tissues, and timescales. It requires cells to move from one part of the body to another, sometimes traveling considerable distances. It requires messages to be sent and received, amplifying or suppressing responses as needed. The messengers of immune communication are cytokines, small proteins released by cells to signal to other cells. There are dozens of different cytokines, each with specific effects. Some promote inflammation, recruiting more immune cells to sites of infection. Some activate particular cell types. Some suppress responses, maintaining balance and preventing excessive reaction. Cytokines can act locally, affecting nearby cells, or systemically, traveling through the bloodstream to coordinate body-wide responses. When you experience fever during an infection, that is partly the work of certain cytokines signaling to your brain to raise your body temperature. Movement of immune cells through the body follows specific pathways. Blood carries immune cells everywhere, but many immune responses happen in tissues outside the blood vessels. Immune cells can exit the bloodstream, squeezing between the cells of vessel walls in a process called extravasation, drawn by chemical signals released at sites of infection or injury. They can also travel through the lymphatic system, a network of vessels that collects fluid from tissues and filters it through lymph nodes, where immune responses are often coordinated. This movement is not random wandering, but directed migration. Immune cells follow gradients of chemical attractants, moving from areas of low concentration toward areas of high concentration. At sites of infection, damaged cells and activated immune cells release these attractants, creating beacons that draw additional defenders. The immune cell feels its environment through receptors on its surface, senses which direction the signal is strongest, and crawls in that direction, sometimes traveling remarkable distances to reach where it is needed. The barriers of the body form the first line of defense, and they are more sophisticated than simple walls. Your skin is both a physical and chemical barrier. The outer layer of dead, flattened cells provides a tough surface that most microorganisms cannot penetrate. The skin also produces antimicrobial peptides, small proteins that can kill bacteria and fungi, and maintains an acidic pH that inhibits many pathogens. The beneficial bacteria that normally live on your skin occupy space and resources, competing with potential invaders, providing protection through their mere presence. Mucous membranes line the respiratory tract, digestive tract, and urogenital tract, all places where the inside of your body meets the outside world. These are not exposed like skin, but are covered with a layer of mucus, a sticky gel that traps microorganisms and foreign particles. In the respiratory tract, tiny hair-like structures called cilia beat rhythmically, moving mucus upward toward the throat where it can be swallowed or expelled. This mucociliary escalator constantly clears the airways, preventing many potential infections from ever taking hold. The digestive tract presents unique challenges for immunity. It must allow nutrients to be absorbed while preventing harmful microorganisms from crossing into the body. It must tolerate the trillions of beneficial bacteria that live in the intestines, bacteria essential for digestion and health. The immune system in the gut is both tolerant and ready. It allows normal gut bacteria to thrive, but responds quickly to pathogens. This balance is maintained through specialized immune structures and constant immune surveillance. When barriers are breached, when skin is cut, when a pathogen evades mucosal defenses, the innate immune system responds immediately. The first cellular responders are often tissue-resident macrophages, large cells whose name means big eaters. Macrophages patrol tissues, sampling their environment through numerous receptors that detect foreign molecules and signs of damage. When they encounter bacteria or dying cells, they engulf them through phagocytosis, enclosing the material in internal compartments where it is broken down by enzymes and reactive chemicals. But macrophages do more than just eat. As they encounter and destroy pathogens, they become activated, changing their behavior and their signaling. Activated macrophages release cytokines that promote inflammation. The redness, warmth, swelling, and pain that mark an active immune response. These cytokines increase blood flow to the area, bringing more immune cells and nutrients. They make blood vessel walls more permeable, allowing fluid and cells to leave the bloodstream more easily. They activate pain receptors, alerting you to the injury or infection. Inflammation, though often uncomfortable, serves important purposes. The increased blood flow brings oxygen and nutrients needed for tissue repair and immune function. The accumulation of fluid dilutes toxins and helps flush them away through the lymphatic system. The recruitment of additional immune cells provides reinforcements for the initial defenders. The changes in tissue environment can also be directly harmful to many pathogens. Some bacteria grow poorly at higher temperatures, for instance, which is part of why fever helps fight infection. Among the first cells recruited to sites of inflammation are neutrophils, the most abundant white blood cells in human blood. Neutrophils are short-lived but potent defenders. They exit the bloodstream rapidly when signaled, moving toward infection with remarkable speed. Like macrophages, they can engulf and kill bacteria through phagocytosis. They can also release their internal contents, enzymes and antimicrobial chemicals, into the surrounding area, creating a toxic environment for pathogens, though potentially damaging to tissue as well. Neutrophils essentially sacrifice themselves in defense, dying after they have exhausted their antimicrobial arsenal. Another early responder is the complement system, not a cell, but a collection of proteins that circulate in the blood in inactive forms. When triggered by bacterial surfaces or by antibodies bound to pathogens, complement proteins activate in a cascade, each active protein activating the next. This cascade results in several defensive effects. Some complement proteins coat pathogens, marking them for destruction by phagocytes. Some directly damage bacterial membranes, creating pores that cause the bacteria to burst. Others serve as chemical signals, attracting immune cells and enhancing inflammation. The complement system amplifies immune responses, providing fast chemical support to cellular defenses. Natural killer cells, often called NK cells, patrol the body looking for cells that have been infected by viruses or have become cancerous. They do not recognize specific pathogens the way adaptive immune cells do. Instead, they recognize when normal cells are stressed or abnormal. Healthy cells display surface molecules that send "I am normal" signals. When these signals are reduced or absent, as happens in some viral infections or cancers, NK cells detect the change and can kill the altered cell before it causes harm. This provides important protection against viruses and cancerous transformation even before adaptive immunity can develop. These innate immune mechanisms, barriers, phagocytic cells, complement, NK cells, operate continuously, providing immediate defense without requiring prior exposure or training. Yet, they are not perfect. Some pathogens evolve ways to evade innate immunity, hiding inside cells where phagocytes cannot easily reach them, producing capsules that prevent complement binding, interfering with normal cell surface signals. When innate immunity alone is insufficient, adaptive immunity takes the stage. bringing specificity and memory to immune defense. The transition from innate to adaptive immunity happens in the lymphoid organs, specialized structures where immune cells develop, mature, and coordinate their responses. The primary lymphoid organs, the bone marrow and thymus, are where immune cells are born and educated. The secondary lymphoid organs, lymph nodes, spleen, and mucosa-associated lymphoid tissues, are where mature immune cells encounter antigens and mount coordinated responses. Bone marrow, the soft tissue inside your bones, is where all blood cells originate, including immune cells. Stem cells in the bone marrow continually divide, generating precursor cells that mature into different cell types. Some become red blood cells, carrying oxygen. Some become platelets, involved in blood clotting. Many become white blood cells, the immune cells we have been discussing. B lymphocytes, one of the two main types of adaptive immune cells, complete their maturation in the bone marrow before entering circulation. T lymphocytes, the other main type, leave the bone marrow early and travel to the thymus to complete their education. The thymus is a small organ located in your chest, just above your heart. It is most active in childhood and gradually shrinks with age, though it continues to function throughout life. The thymus is where T cells learn to recognize self. Developing T cells express receptors that recognize MHC molecules, those identification badges on cell surfaces. These receptors are generated somewhat randomly, creating an enormous diversity of different receptors, but not all are useful. Some T cell receptors would not recognize the body's own MHC molecules at all. These T cells would be useless for defending that particular individual and are eliminated. Other T cell receptors would bind too strongly to self molecules presented by MHC. These T cells could attack healthy tissue and are also eliminated. Only T cells with receptors that bind moderately to self-MHC survive thymic selection, ensuring the T cell population can recognize infected or altered cells but will not attack normal healthy cells under ordinary circumstances. This process of education and selection is remarkable. Of the many T cells developing in the thymus, the majority die without ever leaving. They are tested against the self-antigens present in the thymus and fail the test. The few that pass, those with just the right level of self-recognition, are released into circulation. These survivors form the T cell repertoire, the collection of different T cells available to recognize the countless possible foreign antigens you might encounter. Despite the stringent selection, the surviving T cell population retains tremendous diversity, with each cell carrying a unique receptor capable of recognizing a specific molecular structure. Lymph nodes are small bean-shaped structures located along lymphatic vessels throughout the body. They are dense with immune cells and serve as meeting points where adaptive immune responses are initiated. Lymph fluid, collected from tissues, flows through lymph nodes where it is filtered. Any foreign material, microorganisms, or antigen-carrying cells in the lymph are trapped in the node, where they encounter large numbers of lymphocytes and antigen-presenting cells. If an infection is present in tissue, the draining lymph node often swells as immune cells multiply and responses organize. This is why you might feel swollen lumps in your neck when you have a throat infection. The spleen serves similar functions, but filters blood rather than lymph. It is located in the upper left abdomen and is divided into regions with different functions. The white pulp contains high concentrations of lymphocytes, and is where adaptive immune responses to blood-borne antigens are initiated. The red pulp filters aged or damaged red blood cells from circulation and stores platelets. The spleen is particularly important for defending against pathogens that travel in the bloodstream and for responding to blood-borne antigens that bypass other lymphoid organs. Throughout the body, especially in tissues that interface with the external environment, are collections of lymphoid tissue that do not form distinct organs but serve similar functions. These mucosa-associated lymphoid tissues include the tonsils in the throat, Peyer's patches in the intestines, and scattered aggregates of immune cells in the respiratory and urogenital tracts. They provide local immune surveillance and response capability where the body meets the outside world. Locations where infections often begin. All these structures, bone marrow, thymus, lymph nodes, spleen, mucosa-associated tissues, are connected by the circulatory and lymphatic systems, allowing immune cells to move between them. Mature, naive lymphocytes, those that have not yet encountered their specific antigen, continuously circulate through blood and lymph, passing through secondary lymphoid organs where they might encounter antigens presented by specialized cells. This circulation increases the chances that any given antigen will eventually meet a lymphocyte capable of recognizing it, despite the enormous diversity of possible antigens and the relatively small number of any particular lymphocyte clone in the body at any given time. Antigens are molecules that can be recognized by the adaptive immune system. They are usually proteins or polysaccharides from pathogens, but they can also be other molecules from the environment, from food, or even from one's own body under certain conditions. What makes something an antigen is not an inherent property of the molecule itself, but rather the existence of lymphocyte receptors that can bind to it. The universe of possible antigens is vast. Essentially, any molecular structure that differs from normal self could potentially be antigenic if the right lymphocyte receptor exists to recognize it. The key players in antigen recognition are lymphocytes, B cells, and T cells. Despite their names suggesting different origins, both actually originate in the bone marrow. The B in B cell stands for bone marrow, while the T in T cell stands for thymus, reflecting where each cell type matures. These cells are small, seemingly simple when viewed under a microscope, yet each carries on its surface receptors of extraordinary specificity, capable of distinguishing between molecular structures that differ by as little as a single amino acid. B cells recognize antigens directly through their B cell receptors. These receptors are antibody molecules embedded in the cell membrane. Each B cell produces many copies of a single unique antibody, all with the same antigen binding site. When a B cell encounters an antigen that fits its particular antibody, the antigen binds to many receptors on the cell surface, triggering signaling pathways inside the cell. This can lead to B cell activation, proliferation, and differentiation into antibody-secreting cells or memory cells, processes we will explore more deeply as we continue. T cells recognize antigens differently. They cannot see antigens floating freely in blood or tissue fluid. Instead, T cells recognize small fragments of antigens that are displayed on the surface of other cells, presented by those MHC molecules we discussed earlier. This means T cells can detect what is happening inside other cells. If a cell is infected by a virus, it will display viral protein fragments on its surface, essentially announcing the infection to any T cell with the right receptor. This system allows T cells to find and respond to intracellular pathogens that might otherwise hide from immune surveillance. There are two main categories of T cells, defined by which type of MHC molecule they recognize. CD8 T cells recognize MHC class I molecules, which are present on nearly all cells in the body. CD4 T cells recognize MHC class II molecules, which are present primarily on specialized antigen-presenting cells like macrophages, dendritic cells, and B cells. This division of labor means CD8 T cells can survey all cells for infection or abnormality, while CD4 T cells interact primarily with immune cells, helping to coordinate and enhance immune responses. The generation of lymphocyte receptor diversity is one of the most elegant solutions to a challenging problem. The human genome contains roughly 20,000 genes, yet the immune system can generate millions or even billions of different antibody and T cell receptor specificities. This diversity arises through a process called VDJ recombination, occurring during lymphocyte development in the bone marrow and thymus. The genes encoding antibody and T cell receptors are not complete single genes, but rather collections of gene segments, variable V segments, diversity D segments, and joining J segments. During lymphocyte development, the DNA is cut and rejoined, randomly selecting one segment from each group and splicing them together. This recombination creates a functional receptor gene unique to that particular cell. Additional diversity is introduced by imprecise joining of segments and by random nucleotide additions at the junctions. The result is that each developing lymphocyte generates a unique receptor. This means that in your body right now, you have billions of B cells and billions of T cells, each with a unique receptor. Most of these receptors will never encounter their matching antigen. They exist as unused potential, part of the vast repertoire maintained against possible future threats. A tiny fraction of lymphocytes will, over your lifetime, encounter antigens they can recognize and will become activated, proliferating and carrying out immune functions. An even smaller fraction will persist as memory cells, maintaining long-term protection against specific pathogens. The specificity of lymphocyte receptors explains why adaptive immunity takes days to develop after infection. When a pathogen first enters the body, the number of naive lymphocytes capable of recognizing it is very small, perhaps only a few cells among billions. These rare cells must encounter the antigen, become activated, and proliferate, creating a large enough population to mount an effective response. This clonal expansion takes time. Each activated lymphocyte divides repeatedly, creating a clone of identical daughter cells, all with the same receptor, all able to recognize the same antigen. Over several days, a single activated lymphocyte can give rise to thousands of effector cells. This expansion is not unlimited, though. As the response develops, regulatory mechanisms also activate, preventing excessive or prolonged responses that might damage tissue. Some activated lymphocytes differentiate into regulatory cells that suppress immune responses. Activated lymphocytes also express molecules that, when engaged, deliver inhibitory signals, tempering their activity. As the pathogen is cleared and antigen levels decrease, lymphocytes receive less activating signal, and many die through programmed cell death. The response contracts, leaving behind a small population of memory cells, but removing the bulk of the expanded clone. Memory is perhaps the most important feature of adaptive immunity. After an infection resolves, some of the activated lymphocytes persist as memory cells. These cells are long-lived, sometimes surviving for decades. They retain the same receptor specificity as the original clone, but exist in much larger numbers than naive cells and can respond more rapidly upon re-encounter with the same antigen. This is why second infections with the same pathogen are often less severe or even asymptomatic. The memory response mobilizes faster and more forcefully than the primary response, controlling the infection before it can cause significant symptoms. Memory formation is the principle behind vaccination. Vaccines expose the immune system to antigens from pathogens, either killed or weakened versions of the actual pathogen, or purified components of the pathogen, allowing the adaptive immune system to generate memory without causing disease. When later exposed to the real pathogen, the vaccinated individual has pre-existing memory cells that respond rapidly, preventing or reducing illness. This is immunological memory at its most useful. Protection gained from experience, whether that experience was natural infection or artificial immunization. As we conclude this foundation, notice how the immune system emerges as something far more complex than a simple defense force. It is a learning system, constantly educating itself about what exists in your body and what enters from outside. It is a communication network, coordinating responses across vast distances and multiple cell types. It is a memory system, recording experiences and adjusting future responses accordingly. And it is a regulatory system, balancing activation and suppression, inflammation and resolution, response and tolerance. The mechanisms we have explored, innate and adaptive immunity, cellular and molecular components, barriers and surveillance, recognition and response, form the foundation upon which all immune function rests. They are not isolated pieces, but interconnected elements of a unified whole. The barrier that prevents infection works together with the cells that patrol tissues. The innate cell that first encounters a pathogen instructs the adaptive cell that develops specific protection. The antibody that neutralizes a virus enhances the complement system that destroys bacteria. The memory cell that persists after infection relies on signals from non-immune cells to survive and maintain readiness. We have taken our first steps into the landscape of immunology, established our basic vocabulary and concepts. As we continue, we will explore in greater detail how these components work, how they interact, how they can fail, and how they can be harnessed for therapeutic benefit. We will see how the immune system adapts to different types of threats, how it maintains the delicate balance between protection and tolerance, how it changes over the course of life from infancy through old age. We will discover that the immune system is not merely a defender against disease, but a fundamental part of how we exist as complex organisms in a world full of other life forms, some threatening, many beneficial, all part of the ecological context in which we have evolved and continue to exist. We have walked gently through the foundations, the barriers and cells, the recognition and response, the innate readiness and adaptive learning that form the basis of immune function. Now we continue by observing how these elements combine into coordinated responses, how the immune system adapts its strategies to different types of threats, and how the conversation between different immune components creates the sophisticated protection that allows complex life to flourish in a world filled with microorganisms. Let us turn our attention first to the dendritic cell, a cell type whose importance cannot be overstated despite its relative rarity. Dendritic cells are the bridges between innate and adaptive immunity. the messengers that translate the immediate recognition of danger into the specific, lasting responses of adaptive immunity. They exist in small numbers in tissues throughout the body, particularly at interfaces with the external environment, in the skin, in the lining of the respiratory and digestive tracts, anywhere pathogens might first enter. In their resting state, dendritic cells patrol tissues, sampling their environment through multiple mechanisms. They continuously take up material from their surroundings, fluid, debris, any molecules present, processing what they capture and displaying fragments on their surface using MHC molecules. This sampling is not random surveillance, but active investigation. Dendritic cells extend and retract long membrane projections, reaching out to probe their environment, a behavior that gives them their name. Dendrite means tree-like branch. When a dendritic cell encounters signs of infection or tissue damage, bacterial molecules detected by pattern recognition receptors, inflammatory cytokines released by other cells, stressed or dying cells, it undergoes a dramatic transformation. It stops sampling and begins migrating, leaving the tissue and traveling through lymphatic vessels to the nearest lymph node. As it travels, it matures, changing its surface molecules and its behavior. By the time it reaches the lymph node, it has become specialized for a single purpose, presenting the antigens it captured to T cells. This journey and transformation serve a crucial function. T cells circulate through lymph nodes constantly, but they cannot easily enter tissues to sample antigens directly. Dendritic cells solve this problem by bringing antigens from tissues to the lymph nodes where T cells gather. More than simply transporting antigens, dendritic cells process and present them in ways that allow T cells to recognize them. They also provide additional signals through surface molecules and secreted cytokines that indicate whether the antigens came from a dangerous pathogen or from harmless material. These additional signals help determine whether T cells become activated or remain quiescent. When a T cell with the right receptor encounters its matching antigen presented by a mature dendritic cell, along with the appropriate co-stimulatory signals indicating danger, activation begins. The T cell stops migrating and forms a prolonged interaction with the dendritic cell, a contact lasting hours during which multiple signals pass between the two cells. The T cell receives information about the antigen, about the context in which it was encountered, about what type of response would be most appropriate. Based on these signals, the T cell begins to proliferate and differentiate into effector cells specialized for particular functions. CD4 T cells, upon activation, can differentiate into several subtypes, each producing different cytokines and supporting different types of immune responses. The subtype that emerges depends on the signals the T cell receives during activation, the cytokines present in the environment, the strength of the activating signal, the specific co-stimulatory molecules engaged. This flexibility allows the immune system to tailor its response to different types of threats. Th1 cells, the first helper T cell subset identified, promote responses against intracellular pathogens, particularly bacteria and parasites that live inside macrophages and other cells. Th1 cells produce interferon gamma, a cytokine that activates macrophages, enhancing their ability to kill the pathogens they have engulfed. Interferon gamma also promotes the production of certain antibody types that are particularly effective at coating pathogens and activating complement. The Th1 response is characterized by cellular immunity, relying primarily on activated macrophages and CD8 T cells rather than on antibodies. Th2 cells promote responses against extracellular parasites and are involved in allergic responses. They produce cytokines including IL-4, IL-5, and IL-13, which promote different aspects of immunity. IL-4 drives B cells to produce IgE antibodies, which bind to mast cells and basophils, sensitizing them to allergens or parasites. IL-5 activates eosinophils, specialized granulocytes effective against certain parasites. The Th2 response is characterized by humoral immunity, emphasizing antibody production and specialized effector cells rather than classical cellular immunity. Th17 cells promote responses against extracellular bacteria and fungi, particularly at barrier surfaces. They produce IL-17 and other cytokines that recruit neutrophils and promote inflammation at mucosal surfaces. Th17 responses are especially important for maintaining barrier integrity and defending against pathogens that might breach skin or mucosal defenses. However, excessive Th17 responses are associated with several autoimmune conditions, illustrating how helpful immune mechanisms can cause disease when dysregulated. Regulatory T cells, often called TREGs, represent a different type of CD4 T cell altogether. Rather than promoting immune responses, they suppress them. TREGs produce anti-inflammatory cytokines like IL-10 and TGF-beta and can directly inhibit other T cells through cell-to-cell contact. They are essential for maintaining self-tolerance, preventing immune responses against the body's own tissues, and for limiting immune responses once infections are cleared. Without adequate TREG function, autoimmune diseases develop. With excessive TREG activity, immune responses to pathogens or tumors may be inappropriately suppressed. This diversity of helper T cell responses illustrates a key principle of adaptive immunity. Context matters. The same initial recognition event, a T cell encountering its antigen, can lead to very different outcomes depending on the circumstances. The immune system reads the context through pattern recognition receptors, through the type of dendritic cell presenting the antigen, through the cytokines already present in the environment. It then selects the response strategy most likely to be effective against the particular type of threat encountered. CD8 T cells follow a different path upon activation. They become cytotoxic T lymphocytes, often abbreviated CTLs, specialized for killing infected or abnormal cells. CTLs recognize antigen presented on MHC class I molecules, which means they can survey all nucleated cells in the body. When a CTL encounters a cell displaying foreign or abnormal antigens, viral proteins from an infected cell, tumor antigens from a cancer cell, it forms a tight contact called an immunological synapse. Through this synapse, the CTL delivers lethal signals to the target cell. It releases perforin, a protein that forms pores in the target cell membrane, and granzymes, enzymes that enter through these pores and activate programmed cell death pathways inside the target. The target cell dies through apoptosis, a controlled form of cell death that minimizes inflammation and tissue damage. The CTL then detaches and can move on to kill additional targets. A single CTL can kill multiple infected cells sequentially, making these cells remarkably efficient executioners. The killing performed by CTLs serves multiple purposes. By destroying infected cells, CTLs eliminate the factories producing new viruses, helping to contain viral infections. By killing tumor cells, CTLs provide surveillance against cancer, destroying abnormal cells before they can proliferate into clinically significant tumors. However, CTL activity must be carefully regulated. excessive or misdirected CTL responses can destroy healthy tissue, as occurs in certain autoimmune diseases or in severe transplant rejection. While T cells are orchestrating cellular responses, B cells are developing the humoral arm of adaptive immunity, antibody production. B cells can be activated through multiple pathways. The simplest is direct recognition of antigen by the B cell receptor, followed by co-stimulatory signals from activated helper T cells. In this pathway, a B cell encounters an antigen that binds to its surface antibodies, internalizes the antigen, processes it, and presents fragments on MHC class II molecules. A helper T cell, recognizing these fragments, provides additional activation signals, including CD40, ligand binding to CD40 on the B cell, and cytokine secretion. This T cell help is crucial for most B cell responses, particularly for generating high-affinity antibodies and long-lived memory. Once activated, B cells undergo remarkable transformations. They proliferate rapidly, creating a clone of cells all carrying the same antibody specificity. Within specialized structures in lymph nodes and spleen called germinal centers, these proliferating B cells undergo two key processes: somatic hypermutation and class switching. Somatic hypermutation introduces random mutations into the genes encoding the antibody's antigen binding region. Most mutations are neutral or detrimental, but some improve the antibody's ability to bind antigen. B cells with improved receptors bind antigen more effectively, receive stronger survival signals, and are preferentially selected to continue proliferating. B cells with worse receptors bind antigen poorly and die. Over days to weeks, this process of mutation and selection increases the average affinity of antibodies produced by the responding B cell population, sometimes by 1,000-fold or more. The antibodies produced late in an immune response bind their target much more tightly than those produced initially. Class switching changes the constant region of the antibody, the part that does not bind antigen but determines the antibody's effector functions. All B cells initially produce IgM antibodies. Through class switching, they can change to produce IgG, IgA, or IgE antibodies instead, all with the same antigen specificity but different functional properties. IgG antibodies are most abundant in blood and tissues, effectively neutralizing toxins and viruses, coating pathogens for phagocytosis, and activating complement. IgA antibodies are secreted into mucus, saliva, tears, and breast milk, providing protection at mucosal surfaces. IgE antibodies bind to mast cells and basophils, triggering allergic responses, but also promoting defense against parasites. The particular class that B cells switch to depends on the cytokines present during activation. Helper T cells direct class switching through their cytokine production. Th1 cells promote switching to certain IgG subclasses. Th2 cells promote switching to IgE and other IgG subclasses, and T cells at mucosal sites promote switching to IgA. This coordination ensures that the antibodies produced are appropriate for the type of threat being addressed. Some activated B cells differentiate into plasma cells, specialized antibody factories. Plasma cells are among the most productive cells in the body. A single plasma cell can secrete thousands of antibody molecules per second, maintaining this output for days or weeks until the cell dies. Plasma cells are filled with endoplasmic reticulum, the cellular machinery for protein synthesis and secretion, and have little room for anything else. They do not divide, do not migrate extensively, but simply produce antibodies in vast quantities. The antibodies secreted by plasma cells circulate in blood and permeate tissues, providing widespread protection. When antibodies bind to pathogens, they can neutralize them directly. Antibodies coating a virus prevent it from attaching to and entering cells. Antibodies binding to bacterial toxins prevent the toxins from interacting with their cellular targets. Antibodies coating pathogens also enhance phagocytosis through a process called opsonization. Phagocytes have receptors for the constant region of antibodies, allowing them to bind and engulf antibody-coated particles much more efficiently than uncoated ones. Additionally, antibodies bound to pathogen surfaces can activate the complement cascade, leading to pathogen destruction through complement-mediated lysis or enhanced phagocytosis. Other activated B cells become memory B cells, long-lived cells that persist after the infection resolves. Memory B cells retain the somatic mutations acquired during the primary response, meaning they produce higher affinity antibodies than naive B cells. They also respond more rapidly to antigen upon re-exposure, differentiating into plasma cells within days rather than weeks. The combination of faster kinetics and better antibodies means secondary responses typically neutralize pathogens before they can cause disease. The antibody response illustrates several key features of adaptive immunity. First, specificity. Antibodies produced during an infection recognize antigens from that particular pathogen with exquisite precision. Second, diversity. The antibody repertoire can recognize an almost unlimited number of different antigens. Third, memory. After infection, persistent antibodies and memory B cells provide lasting protection. Fourth, self-regulation. Antibodies themselves provide negative feedback, inhibiting further B cell activation once sufficient antibody has been produced. As we observe these adaptive responses unfolding, we see how innate and adaptive immunity continuously communicate. Innate immune cells do not simply hold the line until adaptive responses develop. They actively shape adaptive immunity through the cytokines they produce, the co-stimulatory molecules they express, the processed antigens they present. Dendritic cells activated by bacterial molecules promote Th1 responses. Those activated by parasitic products promote Th2 responses. The innate immune system essentially interprets the nature of the threat and instructs adaptive immunity on how to respond. Conversely, adaptive immunity enhances innate immunity. Antibodies greatly increase the efficiency of complement and phagocytosis. Helper T cell cytokines activate macrophages and neutrophils, increasing their antimicrobial activities. CTLs destroy infected cells that might otherwise serve as sanctuaries for pathogens hiding from innate defenses. The two arms of immunity are not sequential but synergistic, each amplifying the effectiveness of the other. This synergy extends to the resolution of immune responses. As infections are cleared, multiple mechanisms activate to wind down inflammation and restore tissue homeostasis. Regulatory T cells expand and suppress effector T cells. Alternatively, activated macrophages produce anti-inflammatory cytokines and promote tissue repair rather than pathogen killing. Specialized pro-resolving mediators, lipid molecules derived from omega-3 fatty acids, actively promote resolution, reducing neutrophil recruitment, enhancing dead cell clearance, and stimulating tissue regeneration. Resolution is not merely the passive ending of inflammation, but an active process, carefully orchestrated to repair damage and restore normal tissue function. The mucosal immune system deserves particular attention as it faces unique challenges. Mucosal surfaces, the lining of the respiratory tract, digestive tract, and urogenital tract, represent the largest interface between your body and the external environment. Every breath brings air filled with particles, microorganisms, and potential allergens into your lungs. Every meal introduces not just nutrients, but bacteria, viruses, plant proteins, and other antigenic material into your digestive tract. The mucosal immune system must defend against genuine pathogens while tolerating harmless material and beneficial microorganisms. This tolerance is not simply the absence of response, but active immune regulation. In the intestines, specialized dendritic cells sample antigens from the gut lumen and present them to T cells in ways that promote tolerance rather than activation. They produce TGF-beta and retinoic acid, molecules that favor the development of regulatory T cells over inflammatory T cell subsets. Regulatory T cells in the gut suppress responses to food antigens and commensal bacteria while allowing responses to pathogens. This balance between tolerance and immunity is continuously maintained, adjusted based on the signals the immune system receives from the gut environment. The gut microbiota, the trillions of bacteria, archaea, fungi, and viruses that inhabit the intestines, plays a crucial role in shaping mucosal and systemic immunity. These microorganisms are not passive residents, but active participants in immune development and function. Molecules produced by gut bacteria influence immune cell development, cytokine production, and the balance between inflammatory and regulatory responses. Animals raised in completely sterile environments without any microbiota develop abnormal immune systems. They have fewer and less functional regulatory T cells, altered antibody production, and increased susceptibility to immune-mediated diseases. The relationship between host and microbiota is symbiotic. The bacteria receive a stable, nutrient-rich environment. The host gains multiple benefits. Bacteria produce vitamins and digest otherwise indigestible plant fibers. They compete with pathogens for space and nutrients, providing colonization resistance. They train and educate the immune system, helping it develop appropriate responses and tolerances. Disruption of this relationship through antibiotics, dietary changes, or other perturbations can have lasting effects on immune function and health. Secretory IgA antibodies are particularly important at mucosal surfaces. They are transported across epithelial cells into the mucus layer, where they bind to bacteria and viruses, preventing them from contacting epithelial surfaces without triggering inflammation. Unlike IgG antibodies, IgA does not activate complement or promote phagocytosis in ways that cause tissue damage. Instead, it neutralizes and excludes pathogens gently, maintaining the barrier between the microbial world in the gut lumen and the sterile tissues beneath the epithelium. A single human produces several grams of IgA daily, more antibody than all other classes combined, reflecting the importance of mucosal defense. IgA antibodies also shape the composition of the gut microbiota. They coat bacteria, affecting their ability to colonize and persist. Beneficial bacteria often escape IgA coating through mechanisms that reduce their immunogenicity, while potential pathogens are more heavily coated and excluded. This selective coating helps maintain a beneficial microbial community while preventing overgrowth of harmful species. The immune system is not simply defending against all bacteria, but actively curating the microbial ecosystem in the gut. The respiratory tract employs similar strategies, with mucus, ciliary clearance, IgA antibodies, and immune cells, providing layered defense against inhaled pathogens. The alveoli, the tiny air sacs where gas exchange occurs are particularly vulnerable and are protected by alveolar macrophages that clear inhaled particles and microorganisms. Respiratory infections that overcome these defenses can trigger strong inflammatory responses, recruiting neutrophils and other immune cells to the lungs. While necessary for controlling infection, this inflammation can impair gas exchange and cause the symptoms of pneumonia and other respiratory diseases. Immune responses in the skin differ from those at mucosal surfaces, reflecting different challenges. Skin is a barrier rather than an interface for exchange, and it is constantly exposed to environmental insults, mechanical trauma, and UV radiation. Specialized immune cells reside in different layers of the skin. Langerhans cells, a type of dendritic cell, in the epidermis. Dermal dendritic cells and macrophages in the dermis. T cells scattered throughout. When skin is wounded, inflammation recruits additional immune cells to prevent infection and promote healing. The balance between pathogen clearance and tissue repair is particularly evident in wound healing, where excessive inflammation can delay healing while inadequate immune responses allow infection. Tissue-resident memory T cells have recently been recognized as an important component of immune defense in skin and mucosal tissues. Unlike circulating memory cells, tissue-resident memory cells remain in the tissues where they were first activated, providing local protection against reinfection. A person who has recovered from a cold sore caused by herpes simplex virus has tissue-resident memory T cells in the skin at the site of infection, ready to respond quickly if the virus reactivates. These cells provide faster protection than circulating memory cells, which must first traffic to the site of infection. As we explore these diverse immune environments and response strategies, a picture emerges of an immune system that is highly adaptable, tailoring its responses to the specific anatomical location, the type of threat, and the broader context of the situation. The immune response to influenza virus in the lungs differs from the response to the same virus in the gut because the lung and gut environments present different constraints and opportunities for immune function. The response to a first infection differs from the response to a second infection because of immunological memory. The response in a healthy individual differs from that in someone with underlying immune dysfunction because of different baseline states and capacities. This adaptability extends to the magnitude and duration of responses. Small infections with few pathogens may trigger only local, limited responses. Larger infections with rapidly replicating pathogens may trigger systemic responses. Fever, malaise, production of acute phase proteins by the liver, mobilization of immune cells from bone marrow. These systemic responses carry metabolic costs and cause the symptoms of illness, but they also enhance immune function and help control infections that threaten to become severe. The inflammatory response, which we have mentioned repeatedly, is worth examining more closely as a fundamental immune mechanism. Inflammation is characterized by 5 classical signs, recognized since ancient times: redness, heat, swelling, pain, and loss of function. Each sign reflects specific underlying processes. Redness and heat result from increased blood flow to the affected area. Swelling results from fluid accumulation as blood vessels become more permeable. Pain results from the activation of pain receptors by inflammatory mediators and by the mechanical pressure of swelling. Loss of function can result from pain, swelling, or tissue damage, though it also sometimes reflects an adaptive response that limits use of an injured or infected body part during healing. Multiple molecular mediators drive inflammation. Histamine, released from mast cells, causes rapid vasodilation and vascular permeability. Prostaglandins and leukotrienes, lipid molecules produced from arachidonic acid, promote inflammation and pain. Cytokines like TNF-alpha and IL-1, produced by macrophages and other cells, activate endothelial cells lining blood vessels, causing them to express adhesion molecules that capture passing immune cells and allow them to exit the bloodstream. Complement fragments attract neutrophils and enhance vascular permeability. Collectively, these mediators coordinate the complex changes in tissue environment that characterize inflammation. Acute inflammation, the inflammation that develops over hours to days in response to infection or injury, is generally beneficial when properly regulated. It contains infections, clears damaged tissue, and initiates repair. Chronic inflammation, inflammation that persists for weeks, months, or years, often becomes problematic. When the immune system cannot eliminate a pathogen or when it responds to persistent stimuli like autoantigen or environmental exposures, ongoing inflammation can damage tissues. Many chronic diseases involve maladaptive chronic inflammation. Atherosclerosis involves inflammation in blood vessel walls. Alzheimer's disease involves chronic neuroinflammation. Many cancers develop in settings of chronic inflammation that promotes tumor growth rather than immunity. The distinction between beneficial and harmful inflammation is not always clear-cut and can shift over time. Early inflammation is necessary to control infection, but if it continues too long or is too intense, it causes collateral damage to healthy tissue. The immune system walks a tightrope, needing sufficient inflammation to defend against threats, but not so much that the defense itself becomes destructive. Multiple regulatory mechanisms have evolved to maintain this balance. Anti-inflammatory cytokines that counteract pro-inflammatory signals, regulatory cells that suppress excessive responses, negative feedback loops where inflammatory mediators induce their own inhibitors. When regulatory mechanisms fail, immunopathology results, disease caused by immune responses rather than by pathogens directly. Autoimmune diseases represent one category of immunopathology where immune responses target the body's own tissues. We will explore autoimmunity in more depth later, but it is worth noting here that autoimmune diseases illustrate the central importance of self-tolerance. The immune system's ability to distinguish self from non-self is not perfect, and regulatory mechanisms actively maintain tolerance throughout life. When these mechanisms fail, the destructive power of the immune system turns inward. Hypersensitivity reactions represent another category of immunopathology where immune responses to otherwise harmless antigens cause tissue damage. Allergies are familiar examples. Responses to pollen, food proteins, or other environmental antigens that trigger mast cell degranulation, inflammation, and sometimes life-threatening anaphylaxis. The immune mechanisms involved in allergies are the same ones that defend against parasites, but they are triggered by inappropriate targets in sensitive individuals. Understanding why some people develop allergies while others do not remains an active area of research involving genetics, environmental exposures during development, microbiota composition, and other factors. The sophistication of immune regulation becomes apparent when we consider how the immune system manages its own cells. Immune cells are powerful and potentially dangerous. They can kill other cells, release toxic chemicals, promote inflammation. They must be carefully controlled to prevent harm. Multiple checkpoints regulate immune cell activation, proliferation, and function. Co-stimulatory molecules must be engaged for T cells to activate fully. Cytokine receptors must be expressed for cells to respond to activating signals. Inhibitory receptors can dampen responses even in activated cells. Activated immune cells express molecules like PD-1 and CTLA-4 that, when engaged, deliver inhibitory signals that limit the cell's function. These inhibitory pathways serve crucial purposes in normal immune function, preventing excessive responses and autoimmunity. However, they also limit anti-tumor immunity, as cancer cells can exploit these pathways to suppress immune responses against them. Recent therapeutic breakthroughs in cancer immunotherapy involve blocking these inhibitory pathways with antibodies, releasing the brakes on anti-tumor T cells and allowing them to attack cancer cells more effectively. The success of these checkpoint inhibitor therapies demonstrates both the power of immune responses when fully unleashed and the importance of inhibitory regulation in normal physiology. As we move through this exploration of immune mechanisms and regulation, we see recurring themes: balance between activation and suppression, specificity combined with flexibility, local responses coordinated with systemic changes, innate and adaptive mechanisms working together, immediate protection and long-term memory. The immune system is not a simple on-off switch, but a dynamic network of interacting components, each influencing others, the whole continuously adjusting to changing circumstances. We have now examined the major cellular and molecular players in immune responses: dendritic cells bridging innate and adaptive immunity, helper T cells orchestrating responses, cytotoxic T cells destroying infected cells, B cells producing antibodies, regulatory cells maintaining balance. We have explored how responses differ in different anatomical locations and how they adapt to different types of threats. We have considered both the protective functions of immunity and the potential for immune responses to cause harm. As we continue, we will delve deeper into specific mechanisms, explore how the immune system develops and changes over the lifespan, examine what happens when immunity fails or becomes dysregulated, and consider how immune knowledge is applied therapeutically. The foundation has been laid, the major systems introduced. Now we can explore their interactions and implications with greater depth and nuance, seeing how this remarkable system of protection and regulation shapes health and disease throughout life. We have journeyed through the foundations and explored the major mechanisms: the cells that patrol and defend, the molecules that signal and destroy, the organs where responses coordinate, the strategies adapted to different threats and tissues. Now we turn to integration, observing how these components weave together into unified responses, how the immune system develops and matures over a lifetime, and how the intricate balance of immunity can shift toward dysfunction and disease. Consider for a moment the complexity we have been describing. Your body contains trillions of cells, each with unique molecular signatures. You are continuously exposed to countless microorganisms and environmental antigens. Your immune system must survey all your cells and all these exposures, distinguishing harmless from dangerous, self from non-self, responding appropriately to genuine threats while maintaining tolerance to everything else. This task would seem impossible if not for the elegant integration of multiple systems working in concert. Integration begins before birth, during fetal development. The immune system does not spring into existence fully formed, but develops through carefully orchestrated stages. Early in gestation, hematopoietic stem cells, the precursors of all blood and immune cells, arise in the yolk sac and later migrate to the fetal liver. which serves as the primary site of blood cell production before birth. Later still, hematopoiesis shifts to the bone marrow, where it continues throughout life. The thymus develops and begins receiving precursor cells that will become T cells, starting the process of T cell education even before birth. Fetal immune development occurs in a unique environment. The fetus is not sterile, but is relatively protected from microbial exposure compared to postnatal life. The immune system developing in utero faces a different challenge. It must not reject the mother, despite inheriting paternal antigens that the maternal immune system recognizes as foreign. Multiple mechanisms maintain this tolerance, including specialized cells in the placenta that suppress local immune responses and the selection of fetal regulatory T cells that prevent reactivity against maternal antigens. The fetal immune system is also shaped by maternal antibodies that cross the placenta, particularly IgG antibodies. These maternal antibodies provide passive protection to the fetus and newborn, covering the window before the infant's own adaptive immunity fully matures. An infant born to a mother who has had measles or has been vaccinated against it receives anti-measles antibodies that provide protection for the first several months of life. This transferred immunity is temporary. Maternal antibodies gradually degrade over weeks to months, but it provides crucial protection during early infancy. Birth represents a dramatic transition for the immune system. The relatively sterile fetal environment gives way to massive microbial exposure. The birth canal itself provides the infant's first major colonization with maternal microbiota. Within hours and days, the infant's skin, mouth, and intestines become colonized with diverse microbial communities. The infant immune system encounters thousands of new antigens, triggering responses that shape immune development. The early postnatal period is critical for immune education. Exposure to microbes and environmental antigens during infancy and early childhood influences the development of tolerance and immunity in ways that have lasting effects. The hygiene hypothesis suggests that reduced microbial exposure in early life due to cleaner environments, antibiotics, or other factors may contribute to increased rates of allergies and autoimmune diseases in developed countries. While the details remain debated, evidence supports the general principle that appropriate microbial exposure during development is important for healthy immune function. Breastfeeding provides continued immune support during this critical period. Breast milk contains IgA antibodies, antimicrobial peptides, immune cells, and numerous factors that shape the infant's immune system and microbiota. Oligosaccharides in breast milk serve as prebiotics, promoting the growth of beneficial bacteria in the infant gut. Cytokines and growth factors in breast milk influence immune cell development. The composition of breast milk is not static, but changes over time and even within a single feeding, adapting to the infant's changing needs. As the infant grows, the immune system continues maturing. Lymphoid organs expand. The repertoire of lymphocytes diversifies through ongoing development in bone marrow and thymus. Immune responses become more robust and more refined. Early exposures to pathogens begin building immunological memory. Vaccinations given during childhood leverage the developing immune system's capacity for memory formation, establishing protection against dangerous diseases before natural exposure occurs. The thymus is particularly active during childhood, producing large numbers of naive T cells. As individuals age, the thymus gradually involutes, shrinks, and becomes less active. Thymic output of new T cells declines, though it continues at reduced levels throughout life. This declining thymic function is one aspect of immunosenescence, the gradual changes in immune function that occur with aging. Older adults have fewer naive T cells and more memory T cells, reflecting a lifetime of antigenic exposures but reduced capacity to respond to novel pathogens. Immunosenescence involves multiple changes beyond reduced thymic output. Innate immune cells from older individuals often show altered function. Macrophages may be less phagocytic, neutrophils less effective at killing pathogens. The balance of pro-inflammatory and anti-inflammatory signals shifts, often toward a state of chronic low-grade inflammation, sometimes called inflammaging. B cell function changes with reduced antibody responses to vaccination and infection. These changes contribute to increased susceptibility to infections, reduced vaccine efficacy, and increased cancer incidence in older adults. Yet, the aging immune system is not simply deteriorating but adapting to changing circumstances. The large memory T cell population in older adults provides protection against pathogens encountered earlier in life. The immune system continues functioning, just with different capacities and priorities. Understanding immunosenescence is important for developing better vaccines and treatments for older populations and for addressing the immune-related aspects of age-related diseases. Pregnancy represents another time when immune function must adapt significantly. The fetus expresses paternal antigens foreign to the mother's immune system, yet it is not rejected as a transplanted organ would be. The maternal immune system must maintain tolerance to the fetus while continuing to defend against infections. Multiple mechanisms enable this. The placenta creates a specialized immune environment with reduced inflammatory responses. Regulatory T cells expand and localize to the maternal-fetal interface. Certain molecules on placental cells inhibit NK cell and T cell responses. Disruptions in these tolerance mechanisms can contribute to complications like recurrent miscarriage or preeclampsia. At the same time, pregnancy involves increased susceptibility to certain infections and altered responses to others. Malaria during pregnancy can be particularly severe, partly because the parasite can sequester in the placenta. Influenza during pregnancy carries increased risk of complications. These heightened susceptibilities reflect the immunological adjustments necessary to maintain pregnancy, illustrating the trade-offs inherent in immune regulation. The immune system also shows daily and seasonal variations in function. Circulating immune cell numbers, cytokine levels, and immune responses vary over the course of the day, following circadian rhythms. These rhythms are coordinated by the biological clock and are entrained by light-dark cycles. Disruption of circadian rhythms through shift work, jet lag, or sleep deprivation can impair immune function, increasing susceptibility to infection. The timing of infections can influence their severity, and the timing of vaccination can influence the magnitude of immune responses generated. The immune system does not operate in steady state, but fluctuates rhythmically, integrated with the body's broader temporal organization. Seasonal variations in immune function and infection susceptibility have been recognized for centuries. Respiratory infections peak in winter in temperate climates, partly due to factors like indoor crowding and viral stability in cold, dry air, but also potentially due to seasonal changes in immune function. Vitamin D levels, influenced by sun exposure, fluctuate seasonally and may affect immune responses. Melatonin production varies with day length and influences immune function. The immune system is responsive to environmental cycles, adjusting its function to changing conditions. Nutrition profoundly influences immunity. Severe malnutrition impairs immune function, increasing infection, susceptibility, and mortality. Protein-energy malnutrition reduces lymphocyte numbers and function. Micronutrient deficiencies of vitamins A, C, D, E, and minerals like zinc and selenium impair specific aspects of immunity. Vitamin A is crucial for mucosal immunity and for appropriate T cell differentiation. Vitamin D influences innate immune function and has immunoregulatory effects on adaptive immunity. Zinc deficiency impairs thymic function and T cell development. Adequate nutrition is necessary for proper immune function, while deficiencies create vulnerabilities. Conversely, overnutrition and obesity also affect immunity, though in different ways. Obesity is associated with chronic low-grade inflammation, with adipose tissue producing inflammatory cytokines. This chronic inflammation contributes to metabolic dysfunction and increases risk for cardiovascular disease, diabetes, and certain cancers. Obesity also alters immune responses to infection. Paradoxically, despite chronic inflammation, obese individuals may have impaired responses to some pathogens and reduced vaccine efficacy. The relationship between metabolism and immunity is complex and bidirectional, with metabolic state influencing immune function and immune responses influencing metabolism. The gut microbiota, which we have touched on previously, deserves deeper consideration for its pervasive influence on immunity. The microbiota is not merely present in the gut, but actively communicates with the immune system through multiple mechanisms. Bacterial metabolites, short-chain fatty acids produced by bacterial fermentation of dietary fiber, for example, are absorbed into the bloodstream and can influence immune cells throughout the body. Butyrate, one such metabolite, promotes regulatory T cell development and has anti-inflammatory effects. Gut bacteria also influence systemic immunity by affecting the development and function of immune cells. Segmented filamentous bacteria found in rodent intestines promote Th17 cell development in the gut, affecting immune responses even in distant tissues. Certain bacterial species influence the balance between pro-inflammatory and regulatory T cell populations. The composition of the microbiota can affect susceptibility to autoimmune diseases, allergies, and even responses to cancer immunotherapy. The gut is sometimes called the largest immune organ in the body, not only because of the immune tissue it contains, but because of its profound influence on immunity throughout the organism. Dysbiosis, disruption of the normal microbial community, can have lasting immune consequences. Antibiotics, while essential for treating bacterial infections, also affect beneficial bacteria. Broad-spectrum antibiotics can dramatically reduce microbiota diversity, allowing opportunistic pathogens to overgrow and altering immune function. Recovery may take weeks or months, and some changes can persist long-term. Judicious use of antibiotics is important, not just for preventing antibiotic resistance, but also for preserving beneficial microbiota and the immune functions they support. The nervous and endocrine systems also communicate extensively with the immune system, creating neuroimmune and endocrine immune connections that allow immune function to be integrated with the body's broader physiological state. Stress provides a clear example. Psychological stress activates the hypothalamic pituitary adrenal axis, leading to cortisol release. Cortisol has multiple immunosuppressive effects. It reduces inflammatory cytokine production, inhibits T cell proliferation, and promotes T-cell death. Acute stress can temporarily enhance some immune functions, perhaps preparing the body for potential injury and infection. Chronic stress, however, tends to suppress immunity, increasing susceptibility to infections and impairing wound healing. The sympathetic nervous system also influences immunity through the release of catecholamines like epinephrine and norepinephrine. Immune cells express receptors for these molecules and alter their function in response. Sympathetic activation can affect immune cell trafficking, cytokine production, and inflammatory responses. The nervous system thus provides rapid modulation of immune function in response to environmental challenges, integrating immune responses with the fight-or-flight response and other physiological adaptations to stress. Conversely, the immune system influences the nervous system and behavior. Inflammatory cytokines produced during infection can act on the brain, inducing sickness behavior. The constellation of symptoms including fatigue, reduced appetite, social withdrawal, and increased sleep that accompany many infections. These behaviors, while unpleasant, may be adaptive, conserving energy for immune responses and reducing disease transmission. Depression shares some features with sickness behavior and involves immune activation, though the relationship between immunity and mood disorders is complex and not fully understood. Sleep and immunity are bidirectionally connected. Sleep deprivation impairs immune function, reducing NK cell activity, T cell proliferation, and antibody responses to vaccination. Conversely, infection and inflammation promote sleep through cytokine effects on the brain. Sleep appears to support immune function, with certain immune processes enhanced during sleep. The relationship is not simply that sleep allows immune function, but that sleep and immunity are integrated processes, each supporting the other. Disrupted sleep impairs immunity. Immune activation alters sleep. Maintaining adequate sleep is an important, though often neglected, component of immune health. Physical activity also influences immunity in complex ways. Moderate, regular exercise is generally beneficial, reducing inflammation and improving immune surveillance. Elite athletes training intensely, however, may experience immune suppression and increased infection susceptibility, particularly upper respiratory infections. The relationship between exercise and immunity follows a J-shaped curve, with moderate activity optimal and both sedentary lifestyle and excessive training associated with immune dysfunction. Exercise influences immune function through multiple mechanisms, including effects on immune cell trafficking, anti-inflammatory pathways, and the stress response. We have explored how the immune system integrates with development, aging, circadian and seasonal rhythms, nutrition, microbiota, the nervous system, endocrine system, sleep, and physical activity. This integration means that immune function cannot be understood in isolation, but must be viewed in the context of the whole organism and its environment. Immune health depends not only on the intrinsic function of immune cells and molecules, but also on factors like adequate nutrition, appropriate microbial exposure, regular sleep, stress management, and physical activity. When this integrated system functions properly, health is maintained through continuous surveillance and response to challenges, but the complexity that enables such sophisticated function also creates opportunities for dysfunction. We have already mentioned some forms of immune pathology: autoimmune diseases, allergies, chronic inflammation. Let us now examine these in greater depth, understanding how the mechanisms of protection can become sources of disease. Autoimmune diseases occur when immune responses target the body's own tissues. The mechanisms we have described for fighting infection: antibodies that bind antigens, T cells that kill infected cells, inflammatory responses that recruit and activate immune cells, are turned against self. The resulting tissue damage can affect virtually any organ system, causing diverse diseases with different clinical manifestations, but a common underlying theme of self-reactivity. Why does autoimmunity occur despite the multiple mechanisms maintaining self-tolerance? The answers are complex and vary among different autoimmune diseases, but several general principles apply. First, central tolerance in the thymus and bone marrow is imperfect. Some self-reactive lymphocytes escape deletion and enter circulation. These are normally controlled by peripheral tolerance mechanisms, regulatory T cells, inhibitory signals, lack of co-stimulation. But if these mechanisms fail, autoimmunity can result. Genetic factors influence autoimmunity susceptibility. Certain MHC alleles are strongly associated with specific autoimmune diseases: HLA-B27 with ankylosing spondylitis, HLA-DR4 with rheumatoid arthritis, particular HLA-DQ alleles with type 1 diabetes. These associations likely reflect the fact that MHC molecules determine which self-peptides are presented to developing T cells during selection, and which antigens are presented to mature T cells during responses. Certain MHC molecules may be more prone to presenting self-antigens in ways that activate rather than tolerize autoreactive T cells. Environmental factors also contribute to autoimmunity. Infections can trigger autoimmune diseases through molecular mimicry, where pathogen antigens resemble self-antigens. Immune responses to the pathogen cross-react with self-tissues. Rheumatic fever, which can follow streptococcal throat infection, involves antibodies against streptococcal antigens that cross-react with heart tissue. Other infections may trigger autoimmunity through different mechanisms, by causing tissue damage that releases sequestered self-antigens, by activating innate immunity in ways that break tolerance, or by altering regulatory mechanisms. Tissue damage from any cause can sometimes initiate autoimmunity by exposing self-antigens in inflammatory contexts. Normally, self-antigens are presented in non-inflammatory settings that promote tolerance. When tissue damage occurs with concurrent innate immune activation, self-antigens may be presented with co-stimulation and inflammatory cytokines, conditions that promote immunity rather than tolerance. This can initiate or perpetuate autoimmune responses, creating a cycle where immune-mediated tissue damage leads to further antigen release and immune activation. Once established, autoimmune diseases often become chronic, with immune responses persisting even when the initial trigger is long gone. Autoreactive lymphocytes proliferate and differentiate into memory cells that maintain the response. Tissue damage creates chronic inflammation and continuous antigen presentation. Epitope spreading can occur, where immune responses initially targeting one autoantigen expand to target other antigens in the same tissue as damage releases additional self-antigens and activates additional lymphocyte clones. Treatment of autoimmune diseases often involves immunosuppression, using medications to reduce immune cell function, cytokine production, or inflammatory responses. Corticosteroids broadly suppress immunity and inflammation. Other immunosuppressive drugs target specific pathways, inhibiting cell proliferation, blocking co-stimulation, depleting specific immune cell types, or neutralizing particular cytokines. Newer biological therapies use antibodies to precisely target molecules like TNF-alpha, IL-6 or B cells, providing more specific immunosuppression with potentially fewer side effects than broad immunosuppression. The challenge in treating autoimmune diseases is suppressing pathologic autoimmunity while preserving protective immunity against infections and cancer. Non-specific immunosuppression increases infection risk and potentially cancer risk. The goal is to restore the balance that has shifted towards self-reactivity, ideally by re-establishing tolerance rather than simply suppressing all immune function. Research continues toward this goal, exploring regulatory T-cell therapy, tolerogenic vaccines, and other approaches that might specifically target autoreactive responses while leaving other immune functions intact. Immunodeficiency represents the opposite category of immune dysfunction, inadequate rather than excessive immune responses. Primary immunodeficiencies are genetic disorders affecting immune system development or function. Hundreds of different primary immunodeficiencies have been identified, each affecting different components of immunity. Some affect innate immunity, defects in phagocyte function, complement deficiencies, impaired pattern recognition. Others affect adaptive immunity, defects in T-cell development, B-cell function, or both. Severe combined immunodeficiency, often called SCID, affects both T and B cell development and is one of the most serious primary immunodeficiencies. Infants with SCID are highly susceptible to infections and usually die in the first year of life without treatment. Treatment typically involves hematopoietic stem cell transplantation, replacing the defective immune system with donor stem cells that can develop into functional immune cells. Gene therapy, replacing or correcting the defective gene, has shown promise for some forms of SCID and represents a potentially curative approach to certain primary immunodeficiencies. Other primary immunodeficiencies are less severe but still increase infection susceptibility. Selective IgA deficiency, the most common primary immunodeficiency, involves low or absent IgA antibodies. Many affected individuals remain relatively healthy, but some experience recurrent respiratory or gastrointestinal infections. Common variable immunodeficiency involves impaired antibody production despite normal B cell numbers and can present at various ages with recurrent bacterial infections. Treatment often involves immunoglobulin replacement therapy, regular infusions of antibodies purified from donor plasma. Secondary immunodeficiencies result from external factors that impair immunity. HIV infection causes acquired immunodeficiency by infecting and destroying CD4 T cells, progressively weakening adaptive immunity. Without treatment, HIV infection advances to AIDS, characterized by severe immunodeficiency and susceptibility to opportunistic infections in certain cancers. Antiretroviral therapy, which suppresses viral replication, has transformed HIV from a fatal disease to a manageable chronic condition in settings where treatment is available. Other causes of secondary immunodeficiency include malnutrition, certain medications, cancer and cancer treatments, and chronic diseases like diabetes and kidney disease. Chemotherapy for cancer often causes temporary immunosuppression by killing rapidly dividing cells, including immune cells. Patients receiving chemotherapy are at increased infection risk during periods of low blood cell counts. Immunosuppressive drugs used to prevent transplant rejection or treat autoimmune diseases intentionally impair immunity, increasing infection risk as a trade-off for therapeutic benefit. The importance of intact immunity is never more evident than in immunodeficiency. Infections that are minor inconveniences in healthy individuals can be life-threatening in immunodeficient patients. Opportunistic pathogens, microorganisms that rarely cause disease in healthy people, can cause severe infections in immunodeficiency. The types of infections that occur often indicate which aspect of immunity is impaired. Bacterial infections suggest antibody or complement deficiency. Fungal and viral infections suggest T cell deficiency. Severe bacterial infections, particularly with encapsulated bacteria, suggest complement deficiency. Hypersensitivity reactions, which we mentioned earlier, warrant deeper exploration as another category of immune pathology. The Gell and Coombs classification divides hypersensitivity into 4 types based on mechanism, though real-world reactions often involve multiple types simultaneously. Type I hypersensitivity is immediate hypersensitivity mediated by IgE antibodies and mast cells. This is the mechanism underlying most allergies and anaphylaxis. Initial exposure to an allergen sensitizes the individual, with allergen-specific IgE antibodies produced and bound to mast cells and basophils. Re-exposure causes allergen to cross-link IgE on mast cell surfaces, triggering rapid degranulation and release of histamine, leukotrienes, and other mediators. These cause vasodilation, increased vascular permeability, smooth muscle contraction, and mucus secretion. The symptoms of allergic reactions. Localized type I reactions include allergic rhinitis from inhaled allergens, food allergies causing gastrointestinal symptoms, and allergic asthma causing airway constriction. Systemic type I reactions, anaphylaxis, involve widespread mast cell degranulation, causing cardiovascular collapse and potentially death if untreated. Anaphylaxis requires immediate treatment with epinephrine, which counteracts the effects of mast cell mediators. Type 2 hypersensitivity involves antibodies binding to cell surface or extracellular matrix antigens, leading to cell destruction or tissue damage. Hemolytic disease of the newborn can occur when maternal antibodies against fetal blood group antigens cross the placenta and destroy fetal red blood cells. Transfusion reactions occur when antibodies in the recipient's blood attack transfused red blood cells bearing incompatible blood group antigens. Some drug reactions involve antibodies against drug-modified cell surface proteins. Certain autoimmune diseases involve type 2 mechanisms. Autoantibodies binding to tissue antigens and causing complement activation and cell destruction. Type 3 hypersensitivity involves immune complexes, antibody bound to antigen, depositing in tissues and causing inflammation. Small immune complexes can circulate and deposit in blood vessel walls, kidneys, joints, and other tissues. Complement activation by deposited immune complexes recruits neutrophils, which release enzymes and reactive oxygen species that damage surrounding tissue. Serum sickness, which can occur after receiving foreign serum or certain drugs, involves type 3 hypersensitivity. Some autoimmune diseases, like systemic lupus erythematosus, involve immune complex deposition contributing to tissue damage. Type IV hypersensitivity is delayed type hypersensitivity mediated by T cells rather than antibodies. It develops over hours to days rather than minutes. Contact dermatitis from poison ivy or nickel is a common example. Small chemical molecules bind to skin proteins, creating modified self-antigens that are recognized by T cells. Activated T cells release cytokines that recruit and activate macrophages, causing inflammation and tissue damage. The tuberculin skin test, used to detect tuberculosis infection, relies on type IV hypersensitivity. Individuals previously exposed to tuberculosis develop T-cell responses that cause local inflammation when tuberculosis antigens are injected into the skin. Understanding these mechanisms of immune pathology guides treatment. Antihistamines block histamine receptors, reducing type I hypersensitivity symptoms. Corticosteroids suppress inflammation in multiple hypersensitivity types. Immunosuppressive drugs can prevent type 2 and 3 reactions in transplantation and autoimmunity. Avoidance of allergens or sensitizing substances prevents hypersensitivity reactions from occurring. Desensitization therapy for allergies involves repeated exposure to small doses of allergen, gradually inducing tolerance and reducing IgE responses. The relationship between immunity and cancer illustrates both immune surveillance and immune evasion. The immune system can recognize and destroy cancer cells through multiple mechanisms. Tumor cells often express abnormal proteins. Mutated self-proteins or oncofetal antigens normally only expressed during development that can be recognized as foreign. Stressed or transformed cells may express ligands for NK cell activating receptors. Dying tumor cells release inflammatory signals that activate innate immunity and promote adaptive responses. Despite these surveillance mechanisms, cancers develop and grow, indicating that tumor cells can evade or suppress immunity. Tumors employ multiple immune evasion strategies. They may downregulate MHC expression, making them less visible to T cells. They may express molecules that inhibit T cell activation or induce T cell death. They may recruit regulatory T cells and myeloid-derived suppressor cells that create an immunosuppressive tumor microenvironment. They may produce factors that impair dendritic cell function, preventing effective T cell priming. Cancer immunotherapy aims to overcome immune evasion and enhance anti-tumor immunity. Checkpoint inhibitor antibodies that block PD-1, PD-L1, or CTLA-4 release brakes on T-cell responses, allowing more vigorous anti-tumor immunity. CAR T-cell therapy involves engineering a patient's T-cells to express receptors that recognize tumor antigens, then reinfusing these modified cells to attack the cancer. Cancer vaccines aim to prime immune responses against tumor antigens. Oncolytic viruses selectively infect and kill tumor cells while also stimulating immune responses. The success of these approaches demonstrates that the immune system can control and eliminate cancer when appropriately activated. Transplantation immunology reveals fundamental principles of immune recognition and tolerance. Transplanted organs and tissues express MHC molecules that differ from the recipient's own, unless donor and recipient are identical twins. The recipient's immune system recognizes foreign MHC as non-self and mounts responses against the transplanted tissue, leading to rejection unless prevented by immunosuppression. Transplant rejection can occur through multiple mechanisms. Hyperacute rejection happens within minutes to hours when pre-existing antibodies against donor antigens are present, typically from prior transfusions, transplants, or pregnancies. These antibodies bind to the transplanted tissue immediately, activating complement and causing rapid tissue destruction. Screening for such antibodies before transplantation helps prevent hyperacute rejection. Acute rejection occurs days to weeks after transplantation and involves T-cell mediated immunity. Recipient T cells recognize donor MHC on transplanted tissue as foreign and mount responses that destroy donor cells. Immunosuppressive drugs suppress this response, preventing acute rejection in most cases. Chronic rejection develops over months to years and involves both immune and non-immune mechanisms. leading to gradual loss of graft function. Chronic rejection remains a major challenge in transplantation, as it is less responsive to immunosuppression than acute rejection. To reduce rejection risk, transplants are matched as closely as possible for MHC type between donor and recipient. The closer the match, the less foreign the transplant appears to the recipient's immune system. For kidney transplantation, living-related donors who share half or all of their MHC genes with the recipient provide better outcomes than unrelated deceased donors. For bone marrow or stem cell transplantation used to treat leukemias and other blood disorders, close MHC matching is even more critical, as mismatched transplants can cause graft versus host disease, where donor immune cells attack the recipient's tissues. The immune system's remarkable capacity for learning, memory, and discrimination has made it a target for therapeutic manipulation beyond transplantation and cancer. Vaccines, which we have mentioned repeatedly, represent the most successful immune-based intervention in medicine, preventing millions of deaths annually from infectious diseases. Modern vaccine development continues advancing, with new platforms like mRNA vaccines enabling rapid development and deployment, as demonstrated during the COVID-19 pandemic. Immunotherapy extends beyond vaccines to include antibody therapies, immune cell therapies, cytokine therapies, and immunomodulatory drugs used to treat infections, cancers, autoimmune diseases, and other conditions. Monoclonal antibodies, antibodies produced by clones of identical B cells, can neutralize toxins, block pathogen entry into cells, target cancer cells for destruction, or modulate immune responses by blocking or activating specific molecules. Hundreds of therapeutic antibodies have been developed, treating conditions from cancer to inflammatory diseases to infectious diseases. As we observe the integration of immunity with the whole organism, the sophisticated regulation of immune responses, and the diverse pathologies that can result from immune dysfunction, we gain appreciation for both the power and the fragility of immune protection. The immune system is robust, providing defense against countless threats over a lifetime, yet it can be disrupted by genetic mutations, infections, environmental factors, or aging. It is precisely targeted, distinguishing among millions of different antigens, yet it can be misdirected towards self-tissues or harmless environmental exposures. It is self-regulating, typically maintaining appropriate balance between activation and suppression. Yet this balance can shift toward pathologic extremes. Understanding immunity requires recognizing these dualities: protection and pathology, specificity and flexibility, autonomy and integration, robustness and vulnerability. The immune system is not a simple defender, but a complex adaptive network that maintains health through continuous interaction with the microbial world, integration with other physiological systems, and adjustment to changing circumstances throughout life. In health, this complexity enables sophisticated protection. In disease, it creates opportunities for dysfunction. In medicine, it provides targets for therapeutic intervention. We have traveled through the architecture of immunity, from barriers and innate defenders to adaptive learning and memory, from development through aging, from health to the various forms of dysfunction. Now, as we approach the completion of this exploration, we synthesize what we have learned, observing how all these elements unite into a single integrated system, how immunity connects to the broader story of life and evolution, and how our growing understanding continues to transform medicine and our relationship with the microbial world. The immune system, when viewed in its entirety, reveals itself as something more than a collection of cells and molecules. It is a distributed intelligence, a learning network that spans the body, constantly gathering information about the molecular landscape within and around you. Every moment, immune cells are reading surface markers on your cells, sampling fluid from tissues, surveying the contents of your blood and lymph. They are comparing what they encounter against learned patterns of self and non-self, normal and abnormal, safe and dangerous. They are communicating their findings to other immune cells, adjusting their behavior based on signals they receive, coordinating responses across vast distances. This intelligence is not centralized in any single organ or cell type, but emerges from countless local interactions. A dendritic cell in your skin does not receive instructions from a command center about how to respond to bacteria it encounters. Instead, it recognizes bacterial molecules through its pattern recognition receptors, becomes activated through intracellular signaling pathways shaped by evolution, migrates to the nearest lymph node following chemical gradients, and presents antigens to T cells using mechanisms encoded in its genome and refined through its developmental history. The appropriate response emerges from these distributed processes, from the interactions of many cells, each following relatively simple rules. Yet the system as a whole exhibits properties that transcend what any individual cell could accomplish. It learns from experience, storing memories of past encounters that persist for decades. It distinguishes self from non-self with extraordinary precision despite the millions of possible antigens it might encounter. It adapts its response strategies to different types of threats and different anatomical locations. It maintains tolerance to beneficial microorganisms while defending against pathogens, sometimes making these distinctions among closely related bacterial species. These system-level properties emerge from the integration of innate and adaptive immunity, from the communication networks linking diverse cell types, from the regulatory circuits that maintain balance. The evolutionary perspective illuminates why the immune system has the particular features we observe. Immunity in some form exists in all organisms. Even bacteria have immune-like systems. CRISPR-Cas, which has been adapted as a revolutionary gene editing tool, evolved as a bacterial defense against viral infection, storing memories of past viral encounters in bacterial DNA. Plants, which lack adaptive immunity as animals know it, have sophisticated innate immune mechanisms that recognize pathogen molecules and mount defensive responses. Invertebrate animals have diverse immune strategies, some remarkably complex despite lacking lymphocytes and antibodies. Vertebrates evolved adaptive immunity based on lymphocytes with highly variable antigen receptors approximately 500 million years ago. This innovation, the ability to generate vast receptor diversity through somatic recombination and to create immunological memory, provided powerful advantages in the evolutionary arms race between hosts and pathogens. Pathogens evolve rapidly, with short generation times and large population sizes allowing quick adaptation to host defenses. Hosts, with longer generation times, cannot rely solely on genetically encoded defenses that might become obsolete within a few generations. Adaptive immunity provides a solution. Each individual can generate diverse receptors and learn from experience, allowing flexible responses to rapidly evolving pathogens. The fundamental challenge that drove immune evolution, distinguishing self from dangerous non-self in a world full of microorganisms, remains the challenge your immune system faces today. The The solutions that evolved: pattern recognition receptors that detect conserved microbial molecules, diverse lymphocyte receptors that can recognize any molecular structure, memory cells that provide rapid secondary responses, regulatory mechanisms that maintain self-tolerance. Are the solutions operating in your body right now? You carry within you the legacy of millions of years of host pathogen coevolution encoded in your genome, expressed in the function of your immune cells. This evolutionary history also explains why the immune system sometimes causes disease. Autoimmune diseases may result partly from the inherent difficulty of perfectly distinguishing self from non-self in a system that must be able to recognize any possible foreign antigen. The mechanisms that provide this breadth of recognition: random generation of diverse receptors, selection for receptors that can bind self-MHC, activation by tissue damage or inflammation, create risks of self-reactivity. Evolution has shaped multiple tolerance mechanisms to minimize these risks, but perfection may be impossible. Some level of autoimmunity may be an unavoidable cost of having an adaptive immune system powerful enough to defend against diverse pathogens. Similarly, allergies may result from immune mechanisms that evolved to defend against parasitic worms, multicellular parasites that were common threats throughout human evolutionary history, but are rare in modern developed environments. The Th2 immune responses, IgE antibodies, mast cells, and eosinophils that compose allergic responses are well suited for detecting and expelling intestinal worms. In the absence of parasitic infections, these mechanisms may be triggered inappropriately by harmless environmental proteins causing allergic disease. Some evidence suggests that reduced parasitic infections in modern environments contribute to increased allergy prevalence, though many factors likely contribute to this complex phenomenon. The evolutionary perspective also informs our understanding of the microbiota and its relationship with immunity. Humans did not evolve in sterile environments, but in constant contact with diverse microbial communities. The immune system evolved not to eliminate all microorganisms, but to manage them, excluding pathogens while tolerating or even encouraging beneficial species. The mechanisms of mucosal immunity we explored: IgA antibodies that exclude without destroying, regulatory T cells that suppress inflammation in the gut, innate immune cells that sample antigens and promote tolerance, reflect evolutionary adaptation to life in a microbial world. This long coevolution between hosts and their microbial communities means that the immune system develops and functions optimally in the presence of appropriate microbial exposure. The hygiene hypothesis and related ideas suggest that reduced microbial exposure in modern environments may contribute to immune dysfunction, increased allergies, autoimmunity, and inflammatory diseases, while good hygiene and sanitation have dramatically reduced infectious disease mortality and remain crucially important for public health. There may be costs to eliminating all microbial exposure. Understanding these trade-offs informs approaches to maintaining immune health in modern environments. The relationship between immunity and other physiological systems, which we explored in discussing integration, also reflects evolutionary adaptation. The connections between the immune system and the nervous system allow immune function to be modulated based on environmental threats and stressors. The connections with the endocrine system coordinate immune function with metabolic and reproductive demands. The circadian regulation of immunity aligns immune surveillance and responses with the daily cycle of activity and rest. When different types of threats might be more or less likely, These integrations were shaped by natural selection to optimize survival and reproduction in the environments where humans evolved. Modern medicine increasingly recognizes and utilizes these evolutionary insights. Evolutionary medicine asks why diseases exist and why bodies are vulnerable to particular health problems, often finding answers in evolutionary trade-offs, mismatches between ancestral and modern environments, or pathogen evolution. Understanding immunity through an evolutionary lens informs vaccine design. Vaccines work because they exploit the immune system's evolved capacity for memory. It informs thinking about autoimmunity and allergies. These may be partly consequences of immune mechanisms that were adaptive in ancestral environments. It informs approaches to the microbiota. Maintaining beneficial microbial communities works with rather than against the evolved relationship between immunity and microbiota. The therapeutic manipulation of immunity has become increasingly sophisticated as understanding has deepened. We have mentioned vaccines, immunosuppressive drugs, antibody therapies, and cell therapies. These represent only part of the growing immunological toolkit. Emerging approaches continue expanding possibilities for enhancing beneficial immunity while suppressing harmful responses. Checkpoint inhibitors in cancer therapy work by releasing natural brakes on T-cell responses. Tumors exploit inhibitory pathways like PD-1 and CTLA-4 that evolved to prevent autoimmunity and limit inflammation. Blocking these pathways with antibodies allows T cells to attack tumors more vigorously. The success of checkpoint inhibitors in treating cancers that were previously incurable demonstrates how understanding regulatory mechanisms can be translated into powerful therapies. However, these therapies also cause immune-related adverse effects, essentially autoimmunity, as the released T cells sometimes attack normal tissues. Managing these toxicities while maintaining anti-tumor efficacy represents an ongoing challenge. CAR T cell therapy represents another frontier in cancer immunotherapy. T cells are collected from a patient, genetically engineered to express chimeric antigen receptors that recognize specific tumor antigens, expanded in culture and infused back into the patient. These engineered T cells can achieve remarkable responses in certain cancers, particularly some leukemias and lymphomas. However, they can also cause severe toxicities including cytokine release syndrome, excessive cytokine production causing fever, hypotension, and organ dysfunction. Continued development aims to make CAR T cell therapy safer, more effective, and applicable to more cancer types. Monoclonal antibody therapies have transformed treatment of many diseases. In cancer, antibodies target tumor cells for destruction or block growth signals. In autoimmune diseases, antibodies neutralize inflammatory cytokines like TNF-alpha, IL-6, or IL-17, or deplete specific immune cell populations. In infectious diseases, antibodies can neutralize toxins or viruses. The development of humanized and fully human monoclonal antibodies has reduced problems with immune responses against the therapeutic antibodies themselves. Engineering antibody fragments and bispecific antibodies that simultaneously bind two different targets expands therapeutic possibilities. Cell therapies beyond CAR T cells are being explored. Regulatory T cell therapy aims to treat autoimmune diseases or prevent transplant rejection by infusing expanded TRAGs that suppress pathologic immune responses. Mesenchymal stem cells, which have immunomodulatory properties, are being investigated for treating inflammatory conditions and promoting tissue repair. induced pluripotent stem cells could potentially be differentiated into immune cells for therapeutic purposes. These approaches remain largely experimental, but illustrate the expanding frontier of immune-based therapies. Gene therapy offers possibilities for treating primary immunodeficiencies and potentially for enhancing immunity in other contexts. Replacing or correcting defective genes that cause SCID or other immunodeficiencies can restore immune function, potentially providing permanent cures. Gene editing using CRISPR-Cas and related technologies enables precise modification of immune cell genomes, opening possibilities for engineering immune cells with enhanced or novel functions. Vaccines continue evolving beyond traditional approaches. mRNA vaccines, which deliver genetic instructions for cells to produce antigens rather than delivering the antigens themselves, enable rapid vaccine development and have proven highly effective. Viral vector vaccines use harmless viruses to deliver genes encoding pathogen antigens. Nanoparticle vaccines can present multiple antigens in organized structures that enhance immune recognition. Therapeutic vaccines aim to treat existing infections or cancers rather than prevent disease. Cancer vaccines use tumor antigens to prime immune responses against established tumors. Therapeutic vaccines for chronic viral infections aim to boost T-cell responses that control persistent viruses. Despite remarkable progress, major challenges remain in immunology and immunotherapy. HIV remains incurable despite effective suppressive therapy, as the virus integrates into host cell DNA and hides from immune surveillance. A true cure would require either eliminating all infected cells or inducing immune responses capable of controlling the virus without continuous medication. Vaccines against HIV have been elusive as the virus mutates rapidly and has evolved multiple immune evasion mechanisms. Malaria, tuberculosis, and many other infectious diseases cause enormous global health burdens and remain difficult to prevent or treat. Malaria parasites have complex life cycles and evolved sophisticated immune evasion strategies. Mycobacterium tuberculosis can survive inside macrophages and establish latent infections that persist for decades. Developing effective vaccines against these pathogens requires overcoming substantial immunological challenges. Antigenically variable pathogens like influenza require updated vaccines each year to match circulating strains. Pandemic preparedness requires the ability to rapidly develop and deploy vaccines against emerging pathogens. Autoimmune diseases remain chronic conditions requiring lifelong treatment in most cases. Current therapies suppress immunity broadly or target specific pathways but rarely restore true self-tolerance. The goal of antigen-specific therapy, selectively tolerizing immune responses to self-antigens while preserving other immune functions, remains largely unrealized. Research continues exploring approaches including tolerogenic vaccines, regulatory T-cell therapy, and strategies to induce lasting tolerance. Aging-associated immune decline, immunosenescence, contributes to increased infection susceptibility, reduced vaccine responses, increased cancer incidence, and chronic inflammation in older adults. Understanding and potentially reversing immunosenescence could improve health and extend health span. Approaches being explored include rejuvenating the aging thymus to restore T-cell production, reducing chronic inflammation, and enhancing responses to vaccination in older individuals. The relationship between immunity and the microbiota continues revealing new complexity. The gut microbiota influences not only local immunity but systemic responses, metabolic health, neurological function, and even responses to cancer therapy. Some cancer patients respond well to checkpoint inhibitors while others do not, and microbiota composition appears to influence these responses. Understanding and potentially manipulating the microbiota to improve therapeutic outcomes represents an active research area. Fecal microbiota transplantation, transferring gut microbiota from healthy donors to recipients, has proven effective for treating certain infections and is being investigated for other conditions. The COVID-19 pandemic illustrated both the power and the limitations of immunological knowledge. Vaccines were developed with unprecedented speed using new platforms, demonstrating the potential for rapid response to emerging threats. Understanding of immune responses to the virus informed treatment approaches. However, the pandemic also highlighted gaps in knowledge: why some people develop severe disease while others remain asymptomatic, how long immunity lasts, how to predict and prevent immune-mediated complications. The heterogeneity of immune responses across individuals remains incompletely understood despite decades of research. This heterogeneity, the fact that immune function varies substantially among individuals, reflects the influence of genetics, developmental history, environmental exposures, microbiota composition, age, sex, metabolic state, and other factors. Two people exposed to the same pathogen may have dramatically different outcomes, one developing severe disease, another remaining asymptomatic. Understanding what determines these individual differences could enable personalized approaches to prevention and treatment, tailoring interventions based on an individual's particular immune profile and risk factors. Systems immunology represents an emerging approach to understanding immunity through comprehensive measurement and computational analysis. Rather than studying one gene, one cell type, or one pathway in isolation, systems approaches measure many components simultaneously: gene expression across all genes, protein levels, cell populations, metabolic states, and use computational methods to identify patterns and interactions. These approaches reveal network properties and emergent behaviors that cannot be predicted from studying components in isolation. Systems immunology is beginning to provide more complete pictures of immune responses and to identify biomarkers that predict disease susceptibility or treatment response. Single-cell technologies now allow measurement of gene expression, surface proteins, and other features in individual cells, revealing heterogeneity within cell populations previously thought to be uniform. These technologies have identified new cell types and states, characterized rare cell populations, and mapped cell differentiation trajectories during immune responses. Combined with spatial technologies that preserve information about where cells are located in tissues, single-cell approaches are creating detailed atlases of immune cells in health and disease. Computational immunology uses mathematical and computational models to understand immune dynamics and predict outcomes. Models of infection dynamics describe how pathogen populations grow and how immune responses control them. Models of immune cell interactions capture how signals between cells coordinate responses. Models of immunological memory explain how vaccine-induced protection wanes over time and when boosting might be needed. These models complement experimental work, generating hypotheses, integrating diverse data, and making predictions that guide future research. As we synthesize this knowledge, the mechanisms of immunity, their integration within the body, their evolutionary origins, their dysfunction in disease, their therapeutic manipulation, certain themes emerge repeatedly. Balance appears as a central concept. Balance between activation and suppression, inflammation and resolution, immunity and tolerance, protection and pathology. The immune system must be powerful enough to defend against diverse threats, but restrained enough to avoid attacking self or overreacting to harmless exposures. Multiple regulatory mechanisms maintain this balance, and disease often results when balance is lost. Specificity combined with flexibility represents another central theme. Lymphocytes achieve extraordinary specificity through their unique receptors, each recognizing particular molecular structures. Yet the system as a whole is flexible, capable of responding to any possible antigen through the diversity of the lymphocyte repertoire. Innate immunity sacrifices specificity for speed, using pattern recognition receptors that detect broad categories of threats. The combination of rapid innate responses and specific adaptive responses provides both immediate protection and targeted, lasting immunity. Communication and coordination enable the distributed immune system to function as a coherent whole. Cytokines, chemokines, cell-cell interactions, and adhesion molecules create networks of communication, linking immune cells across tissues and organs. Information flows continuously, from sites of infection to lymph nodes where responses organize, from innate cells encountering pathogens to adaptive cells developing specific responses, from activated immune cells to the nervous and endocrine systems that modulate immunity based on broader physiological state. Memory stands as one of immunity's most remarkable features. The ability to remember past encounters and respond more rapidly and effectively upon re-exposure provides protection that lasts years or decades. This immunological memory, encoded in long-lived memory lymphocytes and long-lived plasma cells secreting antibodies, represents experiential learning at the molecular level. Each infection leaves traces, memory cells waiting to respond if that particular pathogen returns. Vaccination exploits this capacity, creating memory without requiring natural infection. Integration with the whole organism, with development and aging, with metabolism and nutrition, with the nervous and endocrine systems, with the microbiota, with circadian and seasonal rhythms, reminds us that immunity cannot be understood in isolation. The immune system is not a separate module added to the body, but an integral part of physiology, intimately connected with all other systems. Immune health depends not only on the intrinsic function of immune cells, but on overall health, adequate nutrition, regular sleep, stress management, appropriate physical activity, beneficial microbial exposures. As we reach the end of this exploration, return with me to where we began, to the image of your body as a vast city with intelligent, distributed defenses. We can now see this city in greater detail. The borders and barriers, skin, mucous membranes, epithelial linings, constantly maintained and monitored. The resident security forces, tissue-resident macrophages, dendritic cells, innate lymphoid cells, patrolling and sampling. The communication networks, blood vessels, lymphatic vessels, cytokine signals, connecting all parts. The central coordination sites, lymph nodes, spleen, bone marrow, thymus, where intelligence is gathered and responses organized. The specialized forces, neutrophils rushing to sites of infection, T cells killing infected cells, B cells producing antibodies, regulatory cells maintaining peace. The memory archives, long-lived memory cells scattered throughout, ready to respond to previously encountered threats. This city operates continuously, quietly, largely beneath conscious awareness. Right now, as you rest here, your immune cells are circulating, surveying, responding to countless small challenges that never rise to the level of symptoms. Your barrier surfaces are maintaining their integrity, producing antimicrobial molecules, hosting beneficial bacteria that prevent pathogen colonization. Your innate immune cells are clearing dead cells and debris, responding to minor tissue damage, sampling the environment. Your adaptive immune cells are circulating through lymphoid organs, possibly encountering antigens and initiating responses, possibly receiving signals that maintain their quiescent state. Your regulatory mechanisms are maintaining the delicate balance between immunity and tolerance, between protection and restraint. This continuous activity sustains you without effort, without conscious direction. You do not need to instruct your neutrophils to move towards sites of infection. They follow chemical gradients automatically. You do not need to tell your B cells when to produce antibodies. They respond to appropriate signals from helper T cells and other cues. You do not need to consciously maintain tolerance to food proteins or commensal bacteria. Regulatory mechanisms operate autonomously. The immune system, like the heart that beats and the lungs that breathe, functions through elegant biological mechanisms refined over millions of years of evolution. Yet this automatic function does not mean the immune system is beyond influence. Your choices and circumstances matter. Adequate nutrition provides the building blocks and energy immune cells need. Sufficient sleep supports immune function and allows infection-fighting processes to operate optimally. Stress management prevents chronic activation of stress hormones that suppress immunity. Regular moderate physical activity enhances immune surveillance and reduces harmful inflammation. Maintaining beneficial gut microbiota through diet and judicious antibiotic use supports the development and function of the mucosal immune system. Vaccination provides safe exposure to antigens, building memory without risk of disease. Understanding immunity, even at the broad level we have explored, empowers more informed decisions about health. It reveals why vaccines work and why they are among the most effective medical interventions ever developed. It explains why antibiotics should be used thoughtfully, as they affect not only pathogens but beneficial microbiota. It clarifies why autoimmune diseases require suppressing specific immune responses while trying to preserve overall immune function. It illuminates why immunodeficiency increases infection risk and why people with compromised immunity need special precautions. It contextualizes why aging changes immune function and why older adults benefit from certain vaccines young adults do not need. The science of immunology continues advancing rapidly. New discoveries emerge regularly, new cell types identified, new signaling pathways characterized, new therapeutic approaches developed. The picture we have drawn here, comprehensive as it has been, remains incomplete. Much remains unknown about how immune responses are regulated, why they vary so much among individuals, how best to manipulate them therapeutically, how to restore function when it declines or becomes dysregulated. These gaps in knowledge represent opportunities for future research, questions that will occupy immunologists for decades to come. Yet even with incomplete knowledge, the progress in understanding and manipulating immunity has been profound. A century ago, the adaptive immune system was barely understood. Antibodies had been discovered, but their origin and function remained mysterious. The existence of distinct T and B cells was unknown. the molecular basis of antigen recognition was unimaginable. Immunotherapy meant little more than serum therapy. Today, we understand immunity in exquisite molecular detail. We can measure immune responses with single-cell resolution. We can engineer immune cells and antibodies for therapeutic purposes. We can prevent many infectious diseases through vaccination and treat previously incurable cancers by enhancing immunity. This progress reflects the efforts of countless researchers across more than a century, gradually revealing how immunity works through patient observation, careful experimentation, and creative insight. It reflects the development of new technologies, cell culture, monoclonal antibodies, flow cytometry, genetic engineering, genomic sequencing that enabled questions previously impossible to address. It reflects the integration of immunology with other fields, cell biology, genetics, evolutionary biology, computational biology, creating richer understanding than any single discipline could achieve alone. The practical impact of this knowledge extends beyond medicine into biotechnology, agriculture, and other fields. Understanding immune recognition has enabled development of antibodies as research tools in therapeutics. Understanding immune evasion by pathogens informs vaccine design and drug development. Understanding transplantation immunology enables organ transplantation to save lives. Understanding the microbiota's interaction with immunity is beginning to inform agricultural practices and even environmental management. As you absorb this knowledge, recognize that you have gained more than facts and mechanisms. You have gained a way of seeing, a framework for understanding a fundamental aspect of life. You can now observe immunity operating at multiple scales simultaneously. Molecular interactions between receptors and ligands, cellular behaviors as immune cells move and respond, tissue-level changes during inflammation, systemic responses coordinated across the body, lifetime changes from infancy through aging, evolutionary patterns across species. You can appreciate both the power of immunity, its exquisite specificity, its learning capacity, its memory, and its vulnerabilities, the possibilities for dysfunction, the challenges of maintaining balance, the trade-offs inherent in any solution to the problem of distinguishing self from dangerous non-self. This understanding connects you to the broader human endeavor of understanding life and to the particular historical moment in which we live. We are fortunate to live when immunity can be understood and increasingly manipulated therapeutically. People in previous generations suffered and died from diseases we can now prevent or treat. People in future generations will likely benefit from discoveries not yet made, therapies not yet developed, understanding not yet achieved. We occupy a moment in the ongoing progression from ignorance toward knowledge, from helplessness toward capability, from suffering toward health. Let this knowledge rest lightly yet firmly in your mind. The immune system is at work in you now and will continue working throughout your life. It will encounter countless challenges, most of which it will handle successfully without your awareness. It will occasionally fail, leading to infections or allergic reactions or other problems that come to conscious attention. It will gradually change as you age, becoming less capable in some ways, while perhaps gaining wisdom from accumulated experience in others. It will be influenced by your choices, by your environment, by chance events. You cannot control every aspect of immune function, nor would you want to. Conscious management of every immune response would be impossibly complex and would likely work less well than the autonomous regulation that normally operates. But you can support immune health through lifestyle choices. You can make informed decisions about vaccination and other medical interventions. You can appreciate the remarkable system that protects you and understand more fully what is happening when you do experience illness. As this exploration concludes, notice once more your breath, moving gently in and out, just as it did when we began. Notice your body, supported and at rest. Right now, your immune system continues its quiet work. Cells circulating, barriers maintained, signals exchanged. The balance of immunity and tolerance continuously adjusted. All the mechanisms we have explored are operating in you, not as abstract concepts, but as living processes. Molecular interactions in real cells, chemical signals traveling through real tissues, a distributed intelligence maintaining your health through methods refined over millions of years of evolution and operating through elegant biological principles we have only recently begun to understand. The immune system is your constant companion through life, an ancient inheritance, a remarkable example of biological sophistication, a source of both robust protection and occasional problems, a system we are learning to understand and influence therapeutically. It deserves appreciation, even wonder, for what it accomplishes continuously and unconsciously. It deserves care, as its function depends on overall health and appropriate environmental exposures. It deserves understanding, as knowledge of immunity illuminates fundamental questions about life, self, and our relationship with the microbial world. Rest in the knowledge that you are protected by systems that have been millions of years in the making, that you carry within you the capacity to learn from experience at the molecular level, that your body continuously distinguishes self from non-self and responds appropriately to threats while maintaining tolerance to what is harmless or beneficial. Rest in the knowledge that the immune system, for all its complexity, typically functions well, maintaining health through mechanisms both automatic and adaptive, both ancient and capable of lifelong learning. The story of immunity is ultimately a story about life itself, about the challenges all organisms face in a world full of potential threats, about the solutions evolution has discovered, about the trade-offs inherent in any biological system, about the remarkable capacities that emerge from complex networks of relatively simple components. It is a story about you and how you are protected, about humanity and how we have learned to enhance our natural defenses, about life and how it persists in the face of continuous challenges. Carry this understanding forward, allowing it to inform your perspective on health, disease, and the biological basis of existence. Recognize immunity when you see it mentioned in news or encounter it in healthcare settings. Appreciate the vaccines that protect you and the immune memory they create. Understand autoimmune diseases and immunodeficiencies in deeper context. See infections not just as invading pathogens, but as complex interactions between microorganisms and host immune responses. Recognize that your immune health is part of your overall health, influenced by and influencing many other aspects of physiology. Above all, trust in the system that has sustained life through countless challenges over evolutionary time and that continues sustaining you. The immune system is not perfect. It can fail, become dysregulated, cause disease, but it is remarkably capable, refined through millions of years of selection, operating continuously to maintain the integrity of your body against external threats and internal changes. It is worthy of respect, care, and appreciation for the essential role it plays in health and survival. As we complete this journey through immunology, You have gained insight into one of biology's most sophisticated systems. May this knowledge serve you well, informing your decisions, deepening your appreciation for the complexity of life, connecting you to the broader human endeavor of understanding ourselves and the living world we inhabit.